Adriana, thank you so much. Your deeply personal story humbles and motivates and inspires us. And as Robert said in the beginning, it's a core approach of ours to put patient voices like Adriana's first, because in the long run, the most important metric of success that we're going to have we impacted patients' lives in a positive way? Have we changed the world and reduced the fear of vision loss in patients with diabetes? So it is my great pleasure to kick off our first session this morning talking about translating science to clinical care for diabetic retinal disease. Robert mentioned the scientific roadmap that we've put together, and I'll be focusing primarily on the first and third planks of our phase 1 cure platform that we've discussed. And they've really been framed with the response to barriers to progress that we've identified, including you can't solve a problem you haven't defined, and you can't judge success unless you know what to measure. So we started a few years ago by the very ambitious imagining of, at the time of diabetes onset, a multidimensional holistic scale that looked not only at retinal vascular function, as the ETDRS scale currently does, but also worsening neural function in the context of the biochemical milieu and also the systemic health, and how those all relate to quality of life and visual function we thought would be important components of this scale. I want to acknowledge and give my deep gratitude and thanks to the working groups that have done the heavy lift of what we charged them with, and the working group leads that you see here. And over the last year, they've done a remarkable job combing the literature, performing targeted narrative reviews, which, as Robert mentioned, have now been published in Ophthalmology Science. And we're thrilled to have these out there because we think they can serve as a basis for our work going forward. I'll give you a high-level overview of what the groups have looked at, and I just wanted to start with the acknowledgement that's really exemplified here by the first table in the Systemic Health Working Group paper, talking about levels of evidence for the variables that they looked at. And one of the things we charged the groups with was not just to identify promising variables, but also to tell us where the gaps in knowledge are. And what we found is that there are many gaps in knowledge, and that actually, although our hope had been that we would come out with a basket of variables that we could immediately push into a new updated DRD staging system, there's a lot of work still to be done. So when you look at the evidence levels, I've coded them in color for you here, with level 1A evidence being consistent from randomized controlled trials and observational cohort studies, and then decreasing sort of levels of evidence in yellow and in orange. You'll see that very few have level 1A evidence. So there's a lot more work to be done, particularly as we look for factors that contribute information beyond what we can get in a retinal image and in factors that have a relationship to response to treatment. Our other 5 working groups came up with priority variables that are all listed here in one place. And we asked them, as you may remember, to both indicate for us where on the scale of diabetic retinal disease severity those variables were most relevant, and then also to tell us what the stage was in terms of were they ready for current use or ready for use within the next year or two? Were they promising variables with unmet but defined research needs that could be accomplished within the next 5 years? Or did they have potential, but where they had unmet research needs that the group thought they would need more than 5 years to accomplish? And what this has done is this list of what are mostly biomarkers or surrogate endpoints, proposed surrogate endpoints, we hope now to use as the basis for moving into that third plank, looking at identifying endpoints and validating them. The hope is that we can move many of these biomarkers into validation as surrogate endpoints with associated clinical benefit, clinical endpoints, or our real goal is to find and identify new primary endpoints that will be acceptable from a regulatory standpoint in registration trials, primarily because we've heard so clearly from many of you that one of the biggest barriers to developing new therapies, safer, non-invasive, more efficacious therapies, is the lack of endpoints that can be easily achieved in today's landscape. So we've developed and near finalized 2 study designs over the last year in partnership with DRCR Retina Network. These are multi-center longitudinal observational studies. The primary goal is really to define the ocular, structural, and functional characteristics of people with diabetes over a broad range of diabetes duration and disease severity, both in the natural history of the disease and in eyes undergoing treatment. Important secondary outcomes, we hope, will be to investigate retinal structure-function relationships, investigating the degrees to which measures change with increasing severity of diabetic retinal disease, understanding test-retest variability, correlations between two eyes of the same patient, and finally, again, moving whether these structural and functional measurements can be validated as surrogate clinical or primary endpoints. So when you go back to this list of priority variables that we identified, I'm thrilled to show you this in the studies that we've designed. What I've marked in green here are the structural and functional tests that are included in every study visit that we have in these studies. And what's highlighted in yellow are the molecular pathways that were identified primarily by the basic and cellular mechanisms group as of interest that we will hopefully have the ability to address in the biobanked blood and urine samples that we'll be collecting. It has been a really fruitful year in terms of building collaborations, and it has been a real joy to work within both MTM Vision, which is a highly collaborative group, as well as DRCR Retina Network, which is a consortium of retina sites performing high-quality clinical research in retinal disease, to build collaborations between these organizations. And I'm thrilled to say that as DRCR Retina Network has now taken on the agreement to start to run these studies using the DRCR infrastructure, we've also had wonderful and very promising initial conversations with the European Vision Clinical Research Network, and we are hoping to get some of those sites on board for the natural history study in the year or two to come. So I'm going to introduce in the next few slides just the DRCR Retina Network, because we'll be talking a little bit more about them to come, and this collaboration is such an integral part of what will, I think, make us successful in our ability to identify and validate new endpoints. DRCR Retina Network, for those of you who are not familiar with it, is an NIH-sponsored collaborative network of clinical sites across North America. We're dedicated to what we hope is rigorous and impactful clinical research of retinal diseases. We have sort of 4 key components. We get our support from National Eye Institute with important support from NIDDK. I am the chair for the diabetes studies. My colleague Dan Martin chairs the non-diabetes studies. Adam Glassman, who's here today, heads our JABE Coordinating Center with the operational aspects of our group run by Cindy Stockdale. And Claire Calhoun, who's also here today, is serving as the protocol manager for these 2 studies. We have about 126 clinical sites across North America with about 1,400 members right now. And here's just a list to give you some sense of what the history of the network has been. These are the first 30 studies that have been done in the network over the last 20 years, with a Gantt chart showing the periods of recruitment and follow-up. So we're pretty experienced at this point at running clinical studies in retinal disease. And when you look at a snapshot of the current care for diabetic retinal disease, and here it is in one slide, I'm not going to go through it in detail. I will say, although there have been very important lessons that we've learned from industry-sponsored trials, from other academic-sponsored trials, the evidence for each of these bullet points has come from many of the DRCR studies, and we name them by letter of the alphabet, as you can see here. And so it's been very gratifying to know that many of the DRCR studies have really shaped the way we think about current treatment for DRD. I'm thrilled to say that our clinical studies, the natural history study, the eyes under treatment, are the newest DRCR protocols AR and AS. We're almost through our second alphabet. We've got to figure out what to do after we run through this alphabet. And over the past year, we've worked extensively with a protocol development group that includes both membership from MTM Vision and DRCR to finalize the study protocols. We've identified protocol chairs. So I am thrilled to say that Drs. Mohammed El-Masri and Dr. Liane Labriola will be the study chairs for our natural history study. Dr. Tom Gardner, my co-scientific director here at MTM Vision, will serve as the chair for the Eyes Under Treatment study. And we really look forward to their leadership in these. We've also, through DRCR, successfully applied for breakthrough T1D support, so we're very grateful for that support, which is going to go specifically towards standardizing the data and mapping the data variables to common data elements models. And I have to thank Carrie Getz and Michelle Hribar, who were really instrumental in helping us put together some of that planning and some of the thought process behind it. And we're hoping that this helps us increase the shareability of our data sets down the road. The breakthrough T1D support will also help support laboratory testing, biomarkers of kidney function and inflammation, as well as the biobanking of blood and urine samples. So we're poised to start enrolling these studies in early 2025, and we can absolutely use help in terms of getting the word out. We hope that the folks who are clinicians here will be involved as clinical sites. What else is the road ahead? You know, this session was titled sort of translating science to clinical care. And we're really still a little bit on the early side of the scientific understanding and the foundations of that now. Well, I just want to quote from the systemic health paper, and similar sentiments were expressed in the other working group papers as well. After looking at all the available evidence, the conclusions were really that we need an international network of centers able to draw on data from large, diverse cohorts to quickly and efficiently evaluate the prognostic and predictive benefit of risk factors and imaging technologies. And I will add, and potential new therapies for DRD. And as we sort of have that goal in mind, our clear goals are to build academic, industry, and regulatory partnerships over this next year, to continue to encourage incorporation of our promising variables in major clinical studies and trials, to standardize the variables across data sets in accordance with common data element models, with the goal of promoting sharing of clinical and research data sets across groups, and of course, to ultimately support development and validation of new primary endpoints for DRD. So that's a brief update from what we've done over the last year. And thank you all so much for all the work that you've all been involved in. I'd like to welcome up Aude Couturier. She's a professor at Paris Cité University, and she is also the project lead for the Evered Natural History Study, and she's going to be talking to us about preliminary findings from that Evered Natural History Study. Thank you, Aude.
Thank you, Jennifer, and thank you to all the organizers for inviting me today. It's a great honor for me to present our work. The HEAVY-RED study, HEAVY-RED is for intelligent evaluation of diabetic retinopathy. So this is a multicentric study that is conducted in France, and it's a French government-supported research program. It has been led by our estimated and regretted mentor, Ramin Tadayoni, until a few months ago. And I'm now the great honor and mission to lead this project. So this is a consortium between public and private partners. So the public partners are the French hospitals in Paris, the Paris University, Paris Cité, and as well we have an artificial intelligence lab that is working on developing an algorithm. It's the LAT team, which is in the University of Brittany in France. And our private partner are Adsys and Evolucare, that are companies specialized in imaging processing, and Zeiss for the equipment for the OCTA devices. So the background for Evered study was that the current classification based on the heterogeneous studies in the 90s are only based on indirect signs of the disease, so it's really not very accurate for the advanced stages of the disease, and there is no prediction now of the occurrence of edema, and it's only really validated on treatment-naive eyes. So the objective of Evered study is to develop an AI-assisted expert system, so an algorithm, to replace the current classification, so to integrate both imaging data and clinical data, and to estimate the risk for a patient to develop a complication at 1 year, so either PDR or DME. And so the aim is to allow an individual follow-up for each patient based on his own imaging and his data. So the study protocol has been published already in BMJ Open, and so we have now in France 14 ophthalmology centers, 18 diabetology centers that had recruited the patient. So the data collected are visual acuity, but mainly it's anatomical data that we are getting from a different imaging system. So we are focusing on this structural data mainly. All patients underwent ultra-wide-field color fundus photography on 2 different devices. Ultra-wide-field, usually it's Optos device, but also wide-field Clarus device to have real color images. All patients also has OCT and OCTA on 2 different devices, including the Plex Elite wide-field OCTA. All the images are sent to our French HEAVY-RED Reading Center in Paris, and they are evaluated by 2 graders and 1 super grader. So there is 3 lectures of the images. the clinical data that are collected, our demographics, and the information about the diabetes duration and the main parameter of the blood control. So here is the timeline of the project. So we have started the inclusion in 2020, but because of the pandemic, it's really started in 2021. The last patient have been included in December 2023, and the last follow-up visit will be in the next month, in December 2024. All the images will be finally graded in the reading center, expected in June 2025, so that the algorithm should be ready for commercialization in 2026. So the cohorts that we have included is 3,086 patients. So almost all patients have both eyes imaged, and they are follow-up 1 to 3 years, depending on the year of their inclusion. So it's a little more than 2,000 that will have 1 year follow-up, and less patients will have 2 and 3 years follow-up. So now in the reading center, we have already collected 24,000 images, mainly color photos images have been graded now, but we are now working on the grading of the OCT and OCT-A images, and you can see that up to 3,000 have already been validated, and so the images will be graded until June 2025. So we don't have the full result of the study because it's an ongoing study, but here is a short preliminary result of our cohort. So the mean age is 76 years old. The gender is predominantly female. The Caucasian ethnicity is in 65% of the patient, and we have mainly type 2 diabetes, but 24% of the cohort is type 1 diabetes. This may change depending on the last update of the demographics because all the data are not still full. The mean glycated hemoglobin was around 8%. And so the main part of the cohort will be moderate to severe and PDR, which account for 60% of the cohort, but we also have patients with no DR or mild DR, and we will also have a few patients with already end stages of the disease. So we have started to work on the few subset of eyes to start to have some data about the natural history of the diseases. So here is a work that was performed in the department by Dr. Dennis and Dr. Leboite in only 179 eyes, but we will extend to a larger cohort later on. But we can see already that the 1 year change in non-perfusions is not so important, and it depending on the initial stages of the disease. So if we look at the non-perfusion on a wide field plex elite OCTA 15 by 15 millimeters, we can see that the significant difference between the non-perfusion index is only in the moderate to severe NPDR eyes. And so the capillary loss is quite slow in eyes with early stage of the disease. We have also studied the dynamic of non-perfusion pixel appearance, working on a little more than 1 million new pixels that appeared at 1 year, and to try to see if it's appeared randomly in the eyes of the patient or if it's more determined localization compared to the previous non-perfusion area at baseline. And it appeared that it was much more closer to the initial non-perfusion areas than random pixels. So it seems that non-perfusion did not appear randomly in an eyes with diabetic retinopathy, but rather in areas that were initially affected. So there is this kind of susceptibility of some areas to develop non-perfusion that we want to study more. We have also worked on any non-perfusion threshold that could help to diagnose PDR with a non-invasive wide field OCTA, and also to help the clinician to detect this end stage of diabetic retinopathy disease. And it appeared that the threshold of 12% of non-perfusion index on a wide-field OCTA give a quite good accuracy for the PDR detection, but need also to be completed with more wider field of view images. We also work on developed IA tools to help the segmentation of non-perfusion on OCTA image without need to crop out the low quality area of the images. And our partner of LabTeam, this is IA Lab, also work on different tool development. So the final product of EveryRed is supposed to be an algorithm to predict the complication, but there are also other tools that are developed in the center. This is some ancillary projects that have been performed on the Plex Elite of EveryRed patient, and this study have shown that the different field of view of the images can get information for the different stage of the disease. So 6 by 6 millimeter were highly effective for early stage, while larger field of view give better performance for advanced stage of the diseases. And the hybrid fusion system of both 6 by 6 and 15 by 15 demonstrated a better performance. So we also have other work where we implement the clinical data showing even better performance. So we expected that EveryRed project implementing these different imaging together with the clinical data of the patient will provide good results to better determine the risk of progression to one patient to have PDR or DME in the long term. So I would like to thank you for your attention and thanks the team of EveryRed, of course, and a big shout to Professor Tadayoni that have led the project. If you want to visit the website, or ask any question, I will be happy to answer.
Thank you so much. We're going to hold questions until the panel, but we do want plenty of time for discussion. I'd like to welcome Dr. Malvina Edelman. We are sad that Malvina's travel plans unfortunately had a hiccup, so she wasn't able to join us in person, but she will be joining us virtually. And Dr. Edelman, as we know, is the director of the Office of Health Technology that reviews devices for the ophthalmic side within the Office of Product Evaluation and Quality for the FDA. So I'll let Malvina go ahead and speak.
Thank you very much. And sorry I can't be there in person, but I'm really delighted to join this prestigious group and to share a couple of thoughts about how we can incorporate best science in the regulation of medical products for diabetic retinal disease. All of the medical products around the globe are in 4 categories. All medical products are either drugs, devices, biologics, or combination products. The regulation of medical products varies country to country, but even in the United States, understanding how a particular kind of medical product is regulated is sort of like trying to decide on what planet you're on. Even more complicated, within device regulation, there are different classifications, and depending on the classification, the requirements for the medical products differ. So with all of this complexity, I'd like to summarize how can we go from confusion to clarity. While all of the regulation vary, the underlying principle for everything is good science. And I'm delighted to hear about the enormous progress this group is making towards improving the science for DRD. So 2 mechanisms for communicating FDA's recommendations for product developments come via guidance and standards. Guidance describes regulators' interpretation of a policy of a regulatory issue, and they include preclinical and clinical recommendations. Likewise, standards outline both preclinical and recommended clinical trials, and then each regulatory body around the globe has a formal process for recognizing the standard. And once a regulatory body recognizes it, it becomes what's known as a recognized standard, which means that it can now be used for satisfying a pre-market submission requirement. And all of the recognized consensus standards for all of the medical products and all of the guidance for all medical products are on FDA websites. And I've included a lot of links to try to make my comments relatively short, but all of the information is on FDA's websites. So in the next couple of minutes, I'm going to try to summarize where we are and where can we go with all of the fantastic work that this group has done. In the United States, these are all of the drugs that have ever been approved for diabetic retinopathy. These are the ones that are legally currently being marketed. And I'm sure this is not any news to the audience. What I think might not be clear is currently there are no standards, either national or international, that are recognized by FDA for any of the diabetic retinopathy drugs. Furthermore, there are no final guidance available for any diabetic retinopathy drugs. The only document that has ever been issued on the topic has been a draft guidance, and I'd like to highlight that it was published in 2008. So the science behind the draft guidance is the science that was available in 2008. And that was all of the drugs that are currently on the market have used the endpoints that came from that draft guidance. And so for diabetic macular edema, VA endpoints are improvement in greater or equal to 0.3 logMAR, prevention of loss of greater or equal to 0.3 logMAR. And this can be either in percentage of subjects with improvement or mean difference between groups. Same with clinically relevant change in visual function. They recommend visual field mean of greater or equal to 7 dB in area consisting of at least 5 points, and microperimetry with mean greater or equal to 7 dB in an area consisting of at least 5 points. Likewise, the endpoints for DR are based on the 2008 draft guidance, and VA recommendations are for improvement or prevention of loss on ETDRS scale by at least 3 levels for systemically administered products, 3 levels for bilaterally administered products, 2 levels for unilaterally administered products. Or there's an anatomic endpoints, which are prevention of the loss of photoreceptors, prevention of retinal attachment, prevention of nerve fibers. There's some functional and ability to perform an activity of daily living that was not previously able to be performed, but very little has been done in the drug space on that. So that's drugs in a nutshell. Biologics. There are no approved biologics for diabetic retinopathy on the market in the United States. Furthermore, biologics, there are no specific guidance or standards. There is only one generic guidance document on human gene therapy for retinal diseases, and I have the link on the slide here. And all it does is provide common recommendations for clinical trials in ophthalmology space. Does not have any specific references for diabetic retinopathy. So now we come to devices. For devices, the best way to think about it is 3 buckets: screening, diagnosing, and treatment. For screening, we can think of it as teleretinal imaging devices or AI/ML. So as all of you know, there are many different teleretinal imaging devices utilized for diabetic retinal disease today. But the point I want to make is none of them have indications that are specific for diabetic retinal disease. The only devices that are legally on the market for diabetic retinal disease screening are AI/ML devices, and we currently have 3, IDXDR, IART, and IDS. And the IFUs for these are on the slide, as you can see, and I'm sure all of you are well versed on this. For diagnosis, once again, there's nothing on the market that is legally approved or cleared specific for diagnosing of diabetic retinal diseases. However, there are many devices that are cleared to aid in diagnosis, and these are OCT, OCTA, fundus photography, and fluorescein angiography, ultra-wide fundus photography. For treatment, again, no device is legally approved or cleared specific for DRD treatment. There are many devices cleared for laser photocoagulation in diabetic retinopathy, such as PRP or focal grid lasers. And of course, we have devices for vitrectomy and endotamponades. We don't have any guidance or standards that are specific for diabetic retinal devices. However, we do have a number of relevant standards that I thought I would mention. One of them is DICOM, which is interoperability that I'm sure many of you are familiar with, which is essential for AI/ML and as well as teleretinal screening. We have recognized standards for fundus cameras and recognized standards for endotamponades. How do we put it all back together? As I said, the regulations vary country to country and medical product by medical product in the United States. But the concept is that everything should be based on good science. And that's where the clinical reference standards comes in. We have guidance in the device space, but it's really applicable all across, that basically talks about that clinical reference standard is a best available method for establishing a subject's true status with respect to target condition. And it can be a single method or a combination of methods and techniques, including clinical follow-up. It can evolve over time, and it should be the one used for a particular study when reporting performance measures. So for AI/DR screening devices, we have used in the Center for Devices ETDRS protocols and severity scores. And very often people ask me, how did my group get the only autonomous device on the US market to date? And I always give kudos to ETDRS and AO because we were able to point to that and say that we did have a good clinical reference standard that we could utilize for the studies. So where does that bring us? It's really very exciting to hear about tremendous progress MTM has made to date. And what I'd like to do is to challenge all of you to do something today, which I like to think of backcasting. Everybody always talks about forecasting, but I like to challenge you to think about backcasting. And what backcasting is envisioning today where you want to be in the future for all of the regulatory medical products around the globe. Where would you like to be? Where would you like to be with PROs, with clinical endpoints? And then do something where we go back in time to present and figure out all the little steps that need to be taken between now and the future that we want to envision to make that a reality. One way to approach this, one of many ways, is biomarkers. And I'm sure all of you heard of BEST, which was developed by FDA and NIH to promote consistent use of biomarker terms and concepts. Now, what many people don't realize, though, is that biomarkers can have many different categories. They can be used for monitoring, they can be prognostic, diagnostic, predictive, susceptibility. They can summarize susceptibility risk, can be pharmacodynamic, or be utilized for safety. In US FDA, there are two different pathways to integrate biomarkers into medical product developments. They can be used in a single medical product development program, which is a possibility, but as you can imagine, is a very time-consuming and difficult process, or biomarkers can be established for use in multiple development programs by something called biomarker qualification. And there are biomarker qualification programs, both at CDER, which is a Center for Drugs, as well as CDRH, which is a Center for Devices and Radiological Health. Currently, at CDER, there are no ophthalmic biomarkers. So there are no ophthalmic biomarkers currently qualified for either drugs or biologics. At FDA, we currently have 2 ophthalmic medical device development tools, which is equivalent to biomarkers. One of them is PROWL-SS, which is a patient-reported outcomes with LASIK symptoms and satisfaction. And the other one is AIO-LIS instrument, which is assessment of intraocular lens implant symptoms instruments. Both of these are PROs, which can now be utilized as a qualified tool for future medical product developments. And that brings me to the next concept that I want to make sure this group considers, and I believe you actually have considered it a lot more than many other entities that I have spoken to, which is when you think about clinical outcome assessments, all 4 types of assessments should be considered for future or current clinical trials. These include clinician-reported outcome measures, which is, the traditional way that people have thought about clinical trials. But in addition to that, we need to think of how to combine that with patient-reported outcome measures, with observer-reported outcome measures, and performance outcome measures. Each of these are a valid way to assess the patient's performance and should really be thought of as a combination of true evaluation of a patient as a whole. Because we all know that the good physician treats the disease, but the great physician treats the patient who has the disease. Thank you for your attention.
Malvina, thank you so much. I'd like to invite the panel up. I'd like to invite Dr. Couturier, as well as Michael Abramoff from the University of Iowa, Dr. Jason McEnany from the University of Illinois College of Medicine, and of course, Dr. Sandy Puzinski, who is a representative on the MTM Vision Lay Advisory Committee, but who is a scientific powerhouse in her own right. Let's have the group come up, and we will be happy to sort of open up for questions, for feedback, for pushback. As always. I think, why don't we start as we talk a little bit about this translation of science into clinical care and the need for new endpoints, for new ways for us to get new devices, new drugs into the clinics. Perhaps each of you can comment from your diverse perspectives, talk a little bit about what your perspective is and what are the really key issues that we should be focused on as a group in pushing that forward. Out?
Okay, so thank you, Jennifer. I think that the presentation that we just hear help us to put in perspective what we are doing now. And clearly, we need to also add the patient point of view, and also the word of Adriana were very important for us this morning to add also this perspective in our studies in the future. So on my personal point of view, I think that we have in our study mainly focus on the anatomical point of view and anatomical changes, and now to also add more the functional, as you have already planned to do in the natural history study of the DRCONet is really important. And so we also planned to do so as an ancillary project of Everett.
That's wonderful. Michael, can you comment a little bit on your collaborative community and ophthalmic innovation that you and Malvina are leading, and sort of our potential partnerships with MTM Vision there? I think they've taped over the off and on switch, so it should go on automatically.
So it works? Okay.
They're protecting us from ourselves.
Thanks for having me. And before I start, I did want to say that what Adriana just said about barbaric injections, strikes a note. So absolutely, I will remember that for the rest of my life. We need to do something about that and make it better. And then, and Malvina is listening in as well, and I'm sorry she couldn't be here. But I think as I worked on, I have a conflict of interest, right? I'm digital diagnostics founder. As I worked through the various stages of getting from what is better for patient outcome to doing the science, to actually where the science is done, how do you get it to patient? And especially how do you get it to improve patient outcomes? There's a lot that is typically not considered, which is FDA, regulatory approval, how do you get it fitted into the clinical workflow? We're discussing that this morning with a number of you, as well as reimbursement, which I think is very important as well. And how do you essentially, like Malvina was already rightfully saying, sorry, what did she say? Yeah. Sorry. And so it's really important for MTM, which I consider myself a part of, that, you know, where do we want to go and how do we work our way back for what we need to do currently? Because the timelines are always going to be very long, and it's too long already, and we need to get it to patients as soon as possible. And so I think, for example, do you use functional or structural measures, and how much do they correlate with patients' daily lives and the ability to perform specific tasks? And typically, I think structural measures are easier for patients, quicker to do, typically also cheaper than functional measurements, which may take longer, are subjective. I remember my patients taking visible fields and starting crying 20 minutes in. It's just, and then you do an OCT and it takes 30 seconds. And so can we be smart about correlating today what, you know, any measures we want to use as endpoints later on to make sure that we have the most efficient ones? And the other, of course, thing that we're plagued with is that this is a chronic disease. Especially in the early stages, it takes a long time before it manifests in not being able to drive or not being able to see your children. And so relatively easier treatments are when it's a very late stage, because now you can show immediate improvement. If you have an early stage, you either need proxies like the ED2RS, which is so wonderfully developed, or other proxies, or what we call prognostic standards, because otherwise you need to wait 5 or 10 years whether your intervention actually has a better patient outcome. So I think the smarter we are today, the sooner and the better we can get to better patient outcomes.
Yeah, thank you, Michael. And I think you highlight the importance of also parallel efforts as we're doing this planning and backcasting, so that we're working on things simultaneously as opposed to in serial progression.
Yeah, we will not be alive, any of us, when we do it serially. I agree. Yeah.
If I can just pipe in for a sec.
Yes, absolutely.
I don't know if you can hear me.
We can hear you, Malvina.
Okay, great. Thanks, Michael. I think that's a wonderful summary. And Jenny, I think you just said the key point, which is doing things in parallel. And not only in parallel, but I think the unique things that I'd like to point out about CCOI is I think of it sort of like the unique ability to have a helicopter view of the world. So it's trying to see all of the parallel streams and finding one main river into which they all will come in eventually.
Thank you, Malvina. Nice.
I realize now I haven't said anything about CCOI, so thank you, Malvina, for saying that. And so I think CCOI, I think the main goal is to be proactive rather than reactive, rather than for regulators or maybe even reimbursement waiting for someone to come and say, hey, we want to do this or that, what do you think? Rather be proactive and say, today, how can we collect all the experts in the world and say, these should be the endpoints that we'll agree on, and that will make everyone work much faster and easier.
Jason, you have tremendous expertise in visual function testing, and we've certainly leaned on you heavily to help us develop some of the protocols for the studies that we're doing. Comments from your standpoint, particularly lessons from the IRD world, and I know we're going to have a session on that later, but what can we take away from the work that's already been done?
Yeah, I do come from an IRD background, but we've been working in diabetic retinopathy for now for like a decade. And what I was really struck by when I started working in this field was the singular focus on the vascular abnormalities in the disease. And over the last maybe decade or so, this has begun to shift, and the importance of early neural changes is becoming more realized. So I think developing better early stage markers of neural dysfunction that are function-based, and that we compare with structural measures, of course. I mean, the beautiful structural work that you showed. But I think don't forget about function. I think function is critical to the patients. Really, we need to understand how that's changing.
Great. And Sandy, of course, we welcome your perspective not only as a patient advocate, but also as a scientist. What are the key things that you think we should be really looking towards as we hope to translate some of these foundational advances into clinical care?
Well, I think my role, in MTM Vision is to represent the diabetes community. And, as a mother of a child over, decades now who has truly suffered from progressive diabetic retinopathy, I feel a strong need to better engage patients in all the activities that are underway. I do want to recognize, Jen, that you involved the lay advisors in the protocol development for the clinical trials that are going to be underway. That was a remarkable effort that you made to make sure that we were able to review and discuss with you what your plans were, and that's very meaningful. I encourage you to continue to engage us over the trial period so that we can be informed about the progress of the trial. Perhaps we might be helpful in recruitment efforts. Perhaps we could be more helpful in interpreting patient responses to treatment, side effects, that sort of thing. It would be revolutionary if you could incorporate more lay advisor or even patient perspectives on their experience of being in the trial. I think it's really important, and the FDA has a compendium of ways in which we could better engage patients in clinical trials. I would love to see a sharper focus on early intervention, early screening, a much sharper focus on youth onset diabetes. The paper you recently published is striking. And we need to get the message out there to increase the awareness that young people, young children now, born in the late last decade, they're at risk for developing diabetic retinopathy early, and it's hard to detect. So an early focus on this youth onset group, I think, is just critically important. And I will mention that when my child was diagnosed at 11 months of age, I got involved as an advocate for her and other children, along with others in the group here, Marjorie Perry, Judy Hunt, others, and Chris German's parents. We would have fallen off a cliff to protect our child. And I think we got to find those parents of these young kids today, because they will fall off a cliff to help you find ways to better treat and prevent diabetic retinopathy in their kids.
Yeah. We are super grateful.
Can I just add something?
Yes, Melvina, go ahead.
I was truly moved by those comments. And I just wanted to highlight the importance of patient preference studies, which I think you just so eloquently alluded to. I don't believe a patient preference study with FDA's involvement or was ever done in the DRD space. However, we have done and actually co-sponsored one in the glaucoma space. And we've learned tremendous amount from that study that has actually helped us guide future medical product developments. So that is something else that I don't know if MTM has ever considered, but in light of the comments so eloquently just expressed by the speaker, I do think that patient preference study, which truly summarizes what's important to patients and not just the physicians who are treating the patients, can be revolutionary in DRD space.
Malvina, it's a very timely comment, and we've certainly had some early discussions about patient preference sort of drug development panels and eliciting that kind of feedback from patients. I would actually love to hear more about your experience in the glaucoma field, and what was it that was identified, or what were the key things identified in the patient preference panels that then changed how you thought about devices?
So I can send you later the links to the studies, but what we did is under the CERSI, we conducted a very large study with Johns Hopkins, and we had a study around the US asking what's important to patients in glaucoma space. And it was interesting because IOP was something that was important to patients, but only because the doctors told them that it's important. What was important to them is how to ambulate, how to function, how it impacts their daily activities. And that didn't really come across from the doctors. That came across from the patients. And hence our stronger emphasis on PRO and activities of daily living and the impact they have on the patient's performance.
Terrific. Thank you so much. Questions from the audience. I really do want to solicit participation. And if I don't see questions, I'm going to start calling on people. Quentin, please, go ahead.
Hey, Jenny. Hi, Jenny. In your presentation, you talked about clinically validating an endpoint, and in Malvina's, she talked about biomarker qualification. And are those two synonyms? And is that kind of one of the goals of the protocol AR and AS to generate either the biomarker qualification or the clinically validated endpoint?
So I can speak a little bit to the clinical studies and the goals there, and then I'm going to have Malvina speak to the sort of the best qualifications and that process. You know, I think the goals of the clinical studies, we're looking to start to build an evidence base surrounding these clinical tests that are of interest, right? Including, you know, the visual function tests that we've identified, so that we've got, if nothing else, the normative data, the test-retest variability, so that we can use it as a platform to then start perhaps to engage in this process. Who takes charge of this process and how it develops, I think, is something that we really need, all of us as a community, to figure out. And we need the engagement of CCOI and you know, discussions with FDA and the rest, because it, as far as I can tell, it is a lengthy and a demanding process, and probably is beyond, you know, any one of our groups, but together, hopefully, we can make some headway there. Malvina, do you want to talk about that process and what it's like?
Yes, definitely. Thank you, Jenny. So I think with the work you're doing is what's needed for good science. What's needed next, and I do truly believe that CCOI is uniquely positioned to be the entity that can make that happen, is to have a consensus building across United States, but even more broadly around the globe, about how the clinical science that's available today, not in 2008, but available today, should translate into appropriate clinical endpoints for assessment of medical products for DRD, whether it's for screening, diagnosis, or treatment. And I believe the way to do it is to have a consensus building workshop where we ask very difficult questions with everybody around the table, delineating answers. And then I'm not sure of the name of the individual who asked the question, but as far as the process, so the very first thing you do is you build a consensus. Then you probably put together a consensus paper, you publish it, and then from there you can start the official process of biomarker qualification. But again, the first step would be a consensus building workshop. Then that gets translated to a manuscript which summarizes the consensus, because while the biomarker qualification takes a little time, the regulatory bodies usually start acknowledging the consensus building papers for the medical products reviews that are ongoing, while the qualification process can take a little bit of time. Hopefully that clarified rather than confused you.
Beautiful. Thank you, Malvina. Jason?
Yeah, I think that achieving those endpoints that were described earlier is going to be, well, essentially impossible in early stage diabetic retinopathy. Those patients don't have 3 lines of acuity loss to begin with. So what we could do is follow them over time to preserve the good vision the patients have. But I'm wondering, from the FDA standpoint, would you be willing to look at other functional endpoints?
So again, I think I don't think it's whether we'd be willing to do it or not. I think what you need to do is, that's my concept of backcasting. I think what you need to do is put together a consensus committee that is going to try to come up with the key questions that still remain for scientific consensus building around the different endpoints. So do we want to break up DRD into different stages and then have different endpoints depending, to your point, depending on where the patient is and whether it's a different endpoint for treatment, screening, or diagnosing what it is, and then have a scientifically sound discussion. FDA is not, it's not the TAR. FDA is one of the partners around the ecosystem. And I hope people start thinking of it as such. FDA should be basing all the decisions on good science, and it's everybody in this room who should be able to delineate good science. That's the approach I have taken my whole career. That's how we got the premium IOLs on the market. That's how we got MIGS on the market. It's trying to get everybody who has a stake around the medical product ecosystem together in one room, hear everybody's opinions, and come up with a consensus, because ultimately we all have the same goal, which is to improve patients' lives. And so it's bringing everybody together and trying to figure out where is the science is what's needed.
We're going to take a quick comment from Robert and then a last question from Lou that's very concise.
Thanks. Am I on? Yeah. So I just want to reinforce that you framed very well our thinking as to all the work that we've done to date, which is the current endpoints are not adequate, especially to look at early stage disease. And so we did the updated staging, project to start to identify where the promising endpoints might be. And now we've organized the first clinical studies to start to look at functional variables as well against structural to see what we can tease out that can be then used for this consensus process and get forward, and doing it all in parallel. So we recognize the inadequacy of current endpoints, and that's the point behind the work that we've been doing.
Yeah, that's why this ARAS is so essential. This is going to be a paradigm shift in the field if we can follow these patients over years, because we don't have longitudinal functional data in diabetics. It doesn't exist really in the literature. We have some structural data, but not function. I think it's so needed in the field right now.
And even more, we can just wonder if really in the early stage of the disease, the function will be the good endpoint, because of course we want to preserve the function, but even we want more is that to have so early treatment that can preserve all the anatomy. Why should we wait for the main part of the retina to be destroyed to treat a patient just to have, function difference in the patients? So it will be even better in the future to be able to preserve the retina structure so that we don't wait for any functional loss. So that is more challenging in the early stage, for sure.
Lou, we're going to take your last question quickly, and then we'll close.
Thankfully, it's super simple. This is both for Malvina and I guess for Michael potentially. I just want to confirm that CCOI used to stand for Collaborative Community on Ophthalmic Imaging, and now there's more of a place for visual function with the...
The I stands for innovation now, so we changed that a few months ago. And so now it includes everything having to do with innovation, including treatment.
Okay, wonderful, thanks.
And that's very important for what we do together.
Absolutely, and I think it really opens up the ability for partnerships between MTM Vision and CCOI to start to drive some of these consensus building, workshops, papers, et cetera. All right, thank you so much to the panel. Thank you all to the audience, and we'll turn it over to Patrice Fort.